So if they're not ion channels, what are these metabotropic receptors? Most metabotropic receptors belong to an important family of receptors called G-protein coupled receptors, or GPCRs. This receptor family is enormously diverse. There are over 2,000 GPCR genes in the human genome, each with a different preference for binding different neurotransmitters, hormones, or other types of small molecules. To put that in perspective, that's something like 10% of all genes in the human genome, all coding for GPCRs. So that should give you some sense of how important these things are, and how important the diversity of different kinds of GPCRs are. Interestingly, while there's a huge diversity of GPCRs, outside of their specialized ligand binding sites, GPCRs are actually a remarkably similar class of objects. The GPCR is a transmembrane protein complex. This makes sense, since its job is basically to bind molecules outside the cell and affect some change inside the cell. As such, we see the ligand binding portion of the GPCR in the extracellular domain of the receptor. The G-protein coupled receptor gets its name from what happens inside the cell. The intracellular domain of the GPCR binds a set of 3 proteins, labeled alpha, beta, and gamma. Together, these are the G proteins, so named because they bind the molecule guanosine triphosphate, or GTP. If you've taken other biology courses, you might recognize GTP as one of the 4 building blocks of DNA. GTP, like its cousin ATP, adenosine triphosphate, actually plays a variety of different roles in the cell, including serving as the raw material for DNA and RNA synthesis, and acting as an energy storage molecule. In the context of the neuron, GTP plays an important role in intracellular signaling, as we'll see. So now you know the basic pieces of a GPCR. Let's take a look at how they work. The basic principle is common to all GPCRs. A ligand is bound outside of the cell, and this affects the function of the G proteins inside the cell. For example, when the ligand binds to the GPCR, the alpha subunit can break free from the beta and gamma subunits, and it can in turn interact with other proteins, for instance, activating or inactivating them. Frequently, the activated proteins are enzymes, molecular machines that facilitate some particular chemical reaction. These enzymes can cause a variety of effects within the cell, including the generation of other signaling molecules. These signaling molecules are sometimes called second messengers, since they carry a message, albeit a step or two removed from the original receptor interaction. A particularly important second messenger in the neuron is cyclic AMP, which is generated from adenosine triphosphate, or ATP. You might recognize ATP as yet another building block of our DNA, but also as the most prominent energy source utilized in the human body. ATP is converted to its cyclic form by an enzyme called adenylyl cyclase. Adenylyl cyclase plays a role in a variety of cellular processes, and it's activated by the G protein. Cyclic AMP, the second messenger, now activates another enzyme called protein kinase A, or pKa, which in turn now activates many other proteins, including ion channels that allow flux of charge across the membrane, resulting in excitation or inhibition. From the outside, it looks like a bit of a Rube Goldberg machine, but the cell is an exquisitely regulated bit of molecular clockwork, and the existence of many overlapping signaling cascades allows for a precise kind of molecular logic to unfold. The main advantage of GPCRs over ionotropic receptors is not only their versatility, but also the fact that a few molecules of neurotransmitter that bind to the receptor outside of the cell can exert a large amplified effect inside the cell. In addition, GPCRs can have multiple, most often opposing effects on a signal target. For example, in the autonomic nervous system, the 2 neurotransmitters norepinephrine, or adrenaline, and acetylcholine exert opposing effects on the same end organs. While norepinephrine induces the so-called fight or flight response, acetylcholine induces the so-called rest and digest response. The specificity of the effects is mediated by different GPCRs and their associated intracellular signaling cascades. Another conceptual advantage of second messengers is that they can affect multiple targets at once. For example, CAMP can activate or inactivate ion channels in the cell membrane, metabolites in the cytoplasm, and it can even affect gene expression in the cell's nucleus. Such gene expression changes can have profound effects on the function of the cell, and they can even last over potentially very long timescales. By now, hopefully, you're starting to get a sense of how metabotropic receptors bridge between neuronal signaling and the intracellular molecular life of the cell. These mechanisms are essential for affecting long-term changes in neurons, such as in learning, and we'll explore these in greater depth in a future lesson.